Record.
Good morning, everyone. In a short while, we will start a program by singing our national anthem.
Mapita. Good morning, everyone. I think our audio isn't working, so we will start by singing our national anthem. Can you please lead, Don?
Thank you, Ma'am Eddie Flor. Okay. Handa, awit. Bayang magiliw, perlas na silanganan, alab ng puso, sa dibdib mo'y buhay. Lupang hinirang, duyang ka ng magiting, sa manlulupi. Di ka pasisiin sa dagat at bundok, sa simoy at sa langit mo bukaw. May dinag at tula at awit sa paglayang minamahal. Ang kislap ng watawat mo'y tagumpay na nagniningning. Ang bituin at araw ng kailang pa may di magdidilim. Lupa ng araw na pagpapista, buhay langit sa piling mo. Aming ligaya na pag may mga api, ang mapatay ng dahil sa'yo. Okay, so I'm very sorry for the technical difficulties, no? But yeah, ladies and gentlemen, our esteemed guests and participants from the BARMM region and the Mintal Comprehensive High School. Maayong buntag, mapya mapita, good morning everyone. Welcome to our webinar entitled Unlocking the Code, a journey into the basics of molecular biology and its applications. We are gathered here today to embark on a fascinating exploration of molecular biology and its profound impact on research and education in the BARMM region and as well as in MCHS. My name is Joel Don M. Untalan, and I will be your host for today. For some house rules, before we proceed with our program, here are some house rules. At the end of every talk, we will have a 5-minute open forum where you can ask your questions, and you may also raise your hands if you want to ask your questions live. For this webinar is best viewed in the PC or laptop, as some of the Zoom settings are not compatible with iOS phones and some Android devices. And to receive your certificate of attendance, please ensure that you have filled out the attendance and evaluation forms. The link for the attendance form has already been posted in the chat box, and the evaluation link will be provided at the end of the seminar. And last is kindly set your chats from host to panelist to everyone so that everyone can able to see your chats. Okay. So to set the tone for this incredible journey, we are honored to have our three exceptional leaders with us today, each bringing us a wealth of experience and inspiration to us in this wonderful event. Without further ado, let's begin by welcoming our first director. Our first director is the Director General from the Higher Education Subsector of, from the Basic Education Subsector of MBHTE. And it is one of the subsectors from the Bangsamoro Ministry of Basic and Higher Technical Education. His dedication to advancing basic education has made a significant impact on the educational sector. Let us give a warm welcome to Director General Abdullah L. Salik Jr. And I think um, Doc, I think Chief Apolaya, I think you have a message for in behalf of Director General Salik. Oh, okay, later. But yeah, I think let's move on our uh, next director. Now for our next director, let's hear our message from the Director General from the Higher Education Subsector of the Bangsamoro Ministry of Basic and Higher and Technical Education. His leadership has been instrumental in fostering excellence in higher education in the Bangsamoro Autonomous Region. Let's express our gratitude to Director General Marjuni M. Madi. Sir?
I think DG Madi has a problem with his internet, so he'll be back in a while.
Oh, okay, cool. We'll back in a while for Director General Madi. Yeah. And I think we can move on to our next director who can give us welcome remarks. Now, for this project will not commence if we have not sought assistance from a dear friend who has a great passion for community service, who foster our connections between the academia and the communities. Let's all welcome the director of the Ugnayan ng Pahinungod in UP Mindanao, Mr. Michael A. Gatela. Sir Michael, good morning.
Hello, good morning. Assalamualaikum. Mayong hapon, mayong buntag, and good morning everyone. Magandang umaga. I just would like to request the host to allow me to start to put on my video so you can see me. Makita niyo po ako more than the picture that is there, kung ano po yung difference in the itong live speaking engagement with you this morning. Maraming salamat po, and good morning everyone. I believe everyone is here, both from the basic and higher education of the Bangsamoro Ministry of Basic, Higher, and Technical Education, to our organizers from the Philippine Genome Center, Mindanao, to our distinguished speakers for this afternoon, our director of the PGC Mindanao, Professor Joel Tolentino, and the panelists and speakers, and the team of the ever active PGC Mindanao. Sa lahat po ng mga nandito this morning, BARMM or our special guest from those who are outside BARMM, magandang umaga po sa inyong lahat. On behalf of the Ugnayan ng Pahinungod Mindanao and the Ugnayan ng Pahinungod organizations, the whole UP unit, we are very grateful for allowing us to join you this morning in this preliminary events, because I believe there are a series of follow-up hands-on capacity building sessions that will follow after this, entitled An Introduction to Molecular Biology and Its Application. So, konting kwento, our engagement in BARMM started when the Pahinungod program was revived in 2019. We even participated, Pahinungod even participated in some formal and informal discussion, also including the conversations on the consultations regarding the Bangsamoro Education Code. Fast forward, a series of both informal and formal talks, which leads to the creation of the technical working group for both the University of the Philippines and the Ministry of Basic, Higher, and Technical Education last year, which led to the creation of the Memorandum of Agreement, and which was signed last year, June 2022. So we just turned 1 year the partnership with UP and the Ministry of Basic, Higher, and Technical Education. The MOU was signed by the immediate past president, President Danilo L. Concepcion, and on the side of the Ministry of Basic, Higher, and Technical Education, the MOU was signed by Minister 𝗶𝗏. But on the actual event of the MOU signing, the representatives of both Director Generals and I think deputy ministers from the Ministry of Basic, Higher, and Technical Education were there on that historical event. So shortly after that, even from then on, the partnership bloomed and started. There are significant collaborations that we had first, from the program of yung pinaguhugutan ng isang mandato ng University of the Philippines Mindanao, yung affirmative action, we were able to revive again or to re-establish the testing centers for the UP College admission test with the help of the Office of Admissions. I saw in the audience the administrator of UP Agricultural School, si Sir Nano Datukan. That's the first in UP and South UP to have a testing center for UPCAT. Also in Basilan, Sulu, and portions in Maguindanao. So we were also able to generate applications, UPCAT applicants in that area. So I think the number goes to around 2,000 applicants. Moving on, because there are, under this partnership, both MBHTE and the UP agreed on the partnership on the common ground, which is, I will quote that, to deliver responsive, relevant, and quality education for disadvantaged and marginalized learners in BARMM. So yun yung hugot ng ating partnership with BARMM. For the MBHTE, yung major inspiration nito is the Bangsamoro Education Code of 2021, and for UP, our inspiration here is the UP Charter, the RA 9500, which clearly is mention our commitment to national development and public service. So please allow me, I will just enumerate the initial programs and activities that were outlined for that agreement. Is that first is the offering a graduate certification or extension program in BARMM, capacity building for pre-service and in-service teachers, deployment of Gurong Pahinungod volunteers in BARMM, establishing the Bangsamoro Junior Scientist Program, enhancing the physical education and sports development program, engagement in research, and capacity building in food science and technology, and explore corresponding agency stakeholders in the establishment of a tribal university catering to indigenous peoples, enriching Madaris education, strengthen the culture of education, research in the MBHTE, and lastly, the promotion and cultivation of Bangsamoro history, culture, and artistic tradition. So ganun po kalawak yung agreed programs, which I think this training falls into, kung hindi lang siguro isa or more than one yung ina-address po nito na programs and activities under the MOU. Also, the MOU have several memorandum of agreements on the Gurong Pahinungod and service training, and etong latest natin is the memorandum of agreement with the MBHTE for this training for PGC Mindanao. And kami naman sa ugnayan ng Pahinungod, because this is also aligned with our programs on the teacher development and our support to BARMM initiatives and the affirmative action program, because we believe the capacitation of our teachers will also has an effect also to our learners. And in the basic education, alam naman namin na pinanindigan din ng UP Mindanao and the university yung affirmative action, that initiative to provide access to deserving students coming from the marginalized sectors and also our priority areas in Mindanao. So unti-unti meron na pong initial results, and we are happy that the partnership is expanding, and even in the current administration meron na pong mga expanded initiative, also through a creation of the TWG on the Bangsamoro Development Institute. So haba-haba po ng kwento, and but the end goal of this is this collaboration, all in the spirit of developing or contributing to the education sector in BARMM. So kami po sa Mindanao, it's been an honor for Pahinungod also to know the hugot of the BARMM, which is we are also aware of it, yung mission na No Bangsamoro Child Left Behind. So kaisa po kami doon, kaisa kami sa development ng inyong mga learners, and hanggat we are allowed to do our way and we still have our continued support, we will continue to partner and collaborate with BARMM and its units in wherever locations we will be. So with that, maraming salamat po and good luck everyone to this significant event. And congratulations to us all for this initial activity. Maraming salamat po and magandang umaga po sa ating lahat.
Okay, thank you so much for the message, Director Gatela. And now, but last but not the least, let's hear some insightful messages from our new Executive Director of the Philippine Genome Center, 𝗨හි𝗹𝗲𝗰𝗂𝗋 MD. An advocate of educational outreach programs, she is dedicated in bringing the power of molecular biology to our budding researchers and educators. Let's extend a warm welcome to our Executive Director of the Philippine Genome Center, Dr. 𝗨හි𝗹𝗲𝗰𝗂𝗋. Good morning po, ma'am. Sa
Good
Philippines.
morning. Good morning. Should I open my video or you can open the video?
Yes,
is this?
yes, ma'am. It's okay.
All right, good. All right, so magandang umaga po sa kanilang lahat. It's actually an honor and privilege to be doing these welcome remarks to people like you who have been really dedicated in educating our youth and the future of the country, right? So I joined PGC a month ago. I've been in the United States for the past 32 years, and it's but timely to come back. I had started as a faculty member at the UP College of Medicine, and my exposure to genetics has been way back as a Philippine Science High School in Diliman. So it's really the right time to have Philippine Genome Center extend these learnings to educators. And I think I'm very much in a seat where actually I can explain how it's being applied, because I'm a clinical geneticist and a molecular genetic pathologist practicing for the past 35 years. So I wouldn't belabor the point. I think early exposure of students to the concept of molecular biology goes a long way. It's the concept that actually leads to not only medication and therapy, but also knowledge itself, right? So we are all made up of cells, and practically genetics is the whole blueprint of our being. So I keep it short. Wish you all the best. Thank you for the organizers. I think this is a great venue for us to learn from you, and for the speakers, and also for those participants. Take in, absorb, suck in everything that you want to learn from this. It could be the start of your whole career. As I said, I started as a high school student, majored in genetics in undergrad at UP Los Baños, and then pursued it in medicine, and then went into this specialty where actually it's a very exciting field. And this is like the message that I wanted to bring across. We are going to compete globally. With your knowledge and talent, there's that strength that we could put together and harness whatever experiences we have and solve the problems ourselves, because there's no one who would help us but ourselves. So coming from training from abroad, I think this is the time to come and really help out. So we look forward to a successful webinar and maybe the first and maybe more of it in Mindanao and the whole of the Philippines. Thank you very much and enjoy the seminar.
All right, thank you very much for your inspirational message, Dr. 𝗨𝗪𝗪𝗂𝗋𝗆. And also thank you for your inspirational messages, directors. Your presence here elevates the significance of this webinar. This webinar is a collaboration between the Philippine Genome Center, the Ugnayan ng Pahinungod Mindanao, and the Bangsamoro Ministry of Basic, Higher, and Technical Education, or MBHTE. Today, we have gathered to shine a spot on something truly special, which is the availability and accessibility of scientific tools and equipment which we can use in molecular studies. This is particularly crucial for our budding researchers, and as well as in our teachers and some faculty members in the BARMM region and as well as in the MCHS. And our goal here is simple and yet profound, which is to raise awareness and enhance our appreciation for the scientific tools that paved the way for these groundbreaking discoveries. We believe that by understanding the tools at our disposal, we empower ourselves to push the boundaries of knowledge. And how do we plan to achieve this? Well, through dedicated capacity building initiatives like this webinar. We are committed in providing the knowledge and skills needed to navigate the intricate landscape of molecular studies. It's about equipping our community with the tools they need to unlock the secrets of the life itself. So what can you expect from this webinar? Well, we'll be delving into the basics of molecular biology and explore its applications and shedding light on the tools that we can use to make it all possible. So sit back and get ready for your insights, inspiration, and a deeper understanding of the molecular world. So let me introduce our first speaker. So it's my pleasure to introduce our first speaker who will guide us on the start of our journey with molecular biology. And Alicia Marcel Aquino is one of the supervisor of the Research and Development Division of the Philippine Genome Center, Mindanao. She obtained a program of both Bachelor and Master of Science in Biological Engineering from Mapua University, Manila. During this time, she obtained the full scholarship from the DOST ERDT and TEEP scholarship awarded by the Ministry of Education of Taiwan. Her research interest is on in-depth studies of protein interactions using computational tools. And during her free time, she also acts as a research mentor with her recent advices winning the Region 3 representative for the student category of the 16th Philippine National Health Research Week this year, 2023. Ladies and gentlemen, please welcome Ms. Alicia Marcel Aquino, MSc. Take it away, ma'am.
Hello, can you hear me? Is that better? I can hear some Some echo. Hello? Okay. So for the first 20 minutes or so, we're going to talk about the principles of molecular biology and genomics workflow. I hope that by the end of this talk, you'll have some insights on the principles that you will be exposed to hands-on during your molecular biology camp here at PGC Mindanao. So first, what is molecular biology? So molecular biology is the study of the structure and function of macromolecules. So macromolecules such as proteins and nucleic acids that are the building blocks of life. So molecular biology, the way to study it is that it is a reductionist. So from a bigger part, we tend to study the smaller components of a particular portion in this study so that we can understand its structure and function. So first, we start with the cell. So the cell is the basic unit of life. And during the hands-on training here in PGC Mindanao, our samples, we will get maybe the plant cell or the animal cell or the tissue cells, because that is where we will get the genetic information from. So the cell theory has been devised over the years by three main scientists, as shown in the slide. So Matthias Schleiden and Theodor Schwann devised that all organisms are composed of one or more cells, and that the cell is the structural unit of life. So they form the cellular basis of life, but they've agreed that cells can come from non-cellular organisms, which was later disputed by Rudolf Virchow, wherein cells can only arise by division from a pre-existing cell. So this is the general components of the cell. So in the middle is where we can find the majority of the genetic information that we will obtain later on. This is called the nucleus. So the other components of the cell is the plasma membrane and the cytoplasm. Other sources of genetic material for bacteria may be plasmid, eukaryotes, the mitochondria, and in plants, we may also get genetic information from the chloroplast. So throughout this talk, I know you will hear many unfamiliar words. You will be exposed to a lot of information as early as now, but I hope you will gain insights from this, and you will really get a better grasp of all this information when you have the hands-on training here in PGC Mindanao. Then from the cell, we move on to smaller molecules. This is the biomolecules. So we have 4 biomolecules: the lipids, nucleic acids, carbohydrates, and proteins. So the lipids, briefly, they make up our cellular membranes. The nucleic acids store our genetic information. Example of these are the DNA and RNA. Carbohydrates give us energy, and proteins are important in many cellular processes. So the nucleic acids, DNA and RNA, start as a nucleotide. The DNA and RNA differ in the sugar that is attached to them. So in DNA, deoxyribose is the sugar, and in RNA, ribose is attached. Furthermore, this is the difference between DNA and RNA. DNA, as we all know, is double-stranded. RNA is single-stranded. They differ in the bases. So later on, these letters are what we call the bases, and as well as this one. So in RNA, uracil is the equivalent of thymine, because later on in the central dogma, we will see that uracil is more stable in the location where RNA will be produced to protein. Very, very, very technical. So this central dogma is as follows. Really, this is the principle, the central dogma is the principle wherein we synthesize or we make proteins from DNA. So RNA can also undergo replication, such as those in viruses. So if you recall, viruses hijack the molecular or the cell machinery of the host cell. So the RNA will take over the machinery of the host and convert its RNA to DNA. Then the central dogma still can proceed. So this is the difference of the genome and the gene. So you can think of it as gene are the chapters and genome is the book. So the genome contains all these genetic information and gene is just one particular information in this book. So the gene is also known as a DNA sequence. And as I've said, this is also how genes or the DNA sequences are regulated in eukaryotes, similar to the central dogma. So the replication of DNA to RNA happens inside the nucleus of the cell, and outside happens the synthesis of proteins from RNA. So this is the workflow of molecular biology. So we start with nucleic acid extraction until analysis through using bioinformatics or using computer tools. Common applications involve detection of pathogens and identification of organisms or barcoding and phylogenetic studies. Again, this is the workflow. We start with nucleic acid extraction. So in nucleic acid extraction, we have to lyse or to break down the different barriers within the cell to get the genetic material. Then we isolate the nucleic acid or the genetic material and it undergoes purification. Then next, after obtaining the genetic material, we have to quantify it. Did we get enough amount already for us to proceed our experiments? We can do this using spectrophotometry. So this is the basic diagram for a spectrophotometer. The cuvette is the sample holder where we will put our extracted DNA, and a light source will pass through this sample and we get this number called the absorbance. And later on in the laboratory, we have certain ratios that will indicate the purity of the sample we obtain. That is for the purity. We have to determine the integrity of the DNA we obtained. That will be through agarose gel electrophoresis. So DNA is naturally negative. So we place the DNA here at the top row. The first column will be our reference ladder because gel electrophoresis shows the different fragments by size. So when we turn on the electric current, since negative is attracted to positive, the DNA fragments will flow towards the positive electrode. And as it flows to the positive electrode, the fragments will tend to flow based on size. So the lighter fragments will flow faster or more towards the positive electrode. And some dyes will be added so that we can visualize it under ultraviolet rays. This is the setup that you will use in the lab for the gel electrophoresis. So our target is to obtain the one on the left side. So this shows that the DNA that we obtained has good integrity. So after gel electrophoresis, we obtained two factors already: good quantity and good integrity of the DNA. Then next, once we establish those two factors, we can proceed to PCR. So PCR is short for polymerase chain reaction technology. It is used to amplify or make many multiple copies of the same gene or of the same DNA sequence. So we have our target DNA, then these cycles are the different amplification cycles where we produce the same DNA sequence. So why are we doing this? This is because we want to determine the organism or the virus wherein this DNA sequence belongs to, and PCR will help us determine that. So this is the PCR that we will use in the lab and the components of the polymerase chain reaction. So just briefly, the amplification cycle contains 4 parts. So starting from the DNA template, remember DNA is the strands through denaturation. This is done at higher temperature to break the bonds. Then at the annealing portion, we attach the primers. So primers are the sequences also of the DNA surrounding the region we want to target. And then during extension portion, this is where the primers will be extended by another component, the DNA polymerase, to form the multiple copies of DNA. So PCR is influenced by 2 factors, temperature and time. And then since PCR is products also are DNA sequences, we can also apply agarose gel electrophoresis to assess its quality. So check if PCR products have the same or expected band size using age. So in another technology of PCR, we can actually use many DNA sequences or many genes. So for example, in this graph, you can see that we have different genes. This is for COVID pala. So we have many genes that we can see in real time where they are amplified. So we use color tags to each gene to show or to see where they are amplified. And then if they are amplified, then it means the virus or the infection or the pathogen causing the infection is present in the sample. Perhaps in the patient sample, it means the patient is infected. So after this, we can proceed to DNA sequencing. So DNA sequencing is the portion where we determine the sequence of the DNA and whether there are certain variations in the sequence, and we can do that using capillary sequencing. So this is what happens inside a capillary sequencer. You have your sample, and the sample goes through upon introduction of electric impulse, the sample tubes. And once it reaches the detection window, the color tags associated to the different bases, if you recall, the bases are A, G, C, T, they will light up, and then you can determine the sequence as they pass through in a laser. So after all of those experiments, this is the final result. Well, not really the final. We still have to do some quality check, but really this one file is what we need to obtain from all of those experiments. We obtain the DNA sequence. And then we perform some bioinformatics. So bioinformatics, for those that may not know, is the science of collecting and analyzing complex biological data such as genetic codes or the genetic sequence we showed earlier. So if you already have your DNA sequence, you may use different websites to study your DNA sequence, further study it using these websites and using these resources. So I'll show you one example of what we can do with those sequences. This one. So we can do phylogenetic analysis. So you have all these samples and you have identified already the DNA sequence, perhaps for a particular species or the species inside that sample you want to identify. Then we can proceed to identifying the relationship of those species inside your sample using DNA sequencing, using bioinformatics. So after this, as you can see on the screen, we can determine the relationship and the identification of species inside your sample. May it be bacteria or viruses, we can do that. And yeah, in a nutshell, that is what are the principles for bioinformatics and for molecular biology. In a nutshell, this is what you will be exposed to when you take your hands-on training here in PGC Mindanao. And I hope you gained important insights after this lecture. Thank you so much.
All right. Thank you so much, Ma'am Alicia Aquino, for giving us a clear vision of the molecular biology. And now it's time to, before we open our question and answer portion for this, for our first speaker, let me just hear from the message from our Director General, June Madi, who give us a message for this webinar.
Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi ta'ala wa barakatuhu. To our friends from the University of the Philippines Mindanao campus, to the Philippine Genome Center, our participants from the different ministry-supervised HEIs, and other higher education institutions that are participating, to the officials and staff of the BARMM higher education sector who are in attendance to this webinar. A pleasant morning to everyone. I really took time in connecting to the Zoom, but I think the challenge was more of the compatibility of the operating system of my tablet and the upgraded requirements of the Zoom link. So for that, I apologize not having been able to participate on time, but I'm trying my best through this video to reach out to everyone. I would have loved to be physically with the team, but I think there are some limitations now. We just had our regional TSD committee meeting here in Manila, and one of the important discussions were the agriculture sector of our territory. And I think in those discussions, it appeared to me that collaborations in terms of our programs, activities, and to include the strategic directions are gearing towards our investments in the higher education for the agriculture programs. So I think it's just timely that our counterpart, our partners, especially from the PGC of the UP Min, are providing their technical expertise, are providing their resources, and most importantly, their time to share with us. Whatever we can partake of this engagement, it is for the betterment of our studentry, of our schools, and to include our region as a whole. I have no other wishes but for all the participants to learn and to make sure that you have something to bring to your institutions and after this webinar. So, mulit-muli, kami po ay nagpapasalamat sa UP, University of the Philippines, at kami po ay malubos na tumatangkilik sa lahat ng mga aktibidades at mga programa na pwedeng i-share ng isang University of the Philippines doon sa mga higher education institutions na nasa Bangsamoro territory. Maraming salamat po, and I am really sorry that I cannot make it to the best as a panelist. Good morning.
Okay, thank you for that wonderful and motivational message, Director General Maddy. And also, may we also recognize here the presence of our on behalf of Director General Salik, we have here Sir 𝗕𝗀𝗏, who can give us the welcome message for our in behalf of the Director General of the Basic Education. Yeah. Good morning po.
Yes, good morning. Good morning. Assalamualaikum warahmatullahi wabarakatuh. And it's a privilege to be joining you in this webinar. And it's also a great blessing that we are here together. Yeah, in behalf of the Director General for Basic Education, Director General Abdullah Jun Salik Jr., he would like to convey na hindi siya makakasali sa atin dito ngayon for reasons na pinatawag siya ni Minister Val, may emergency meeting sila. So in his behalf, I'd like to say thank you to UP, to the Philippine Genome Center, and to all that are attending in this webinar. Maraming salamat po sa inyo. Just would like to highlight just one thing. Para po sa amin sa BARMM, this is a very important program. I think ito po yung kauna-unahang program na mangyayari po para sa aming mga guro dito sa BARMM. Kinakailangan po namin iangat yung kalidad at antas ng pagtuturo ng science and mathematics. Ito po ay lumabas sa isang pananaliksik na ginawa recently ng UP-ACTRC na ilan sa mga subjects na kailangan tutukan po namin ay ang science and mathematics. Kung kaya't malaking pribilehiyo po para sa amin na makasama po ang mga sayantista ng UP, ng Philippine Genome Center para iangat po ang kakayahan at kaalaman ng aming mga guro sa science and mathematics na nagtuturo ng science and mathematics sa junior science high school. Junior and senior high school. Para naman po sa amin, finally, ito po ay kabahagi din po ng aming Bangsamoro Junior Scientist Program na nilunsad po namin last 2 years ago bilang pagpapaangat at pagpapalaganap ng kultura ng pananaliksik. At kasabay dito, of course, yung pag-angat ng kaalaman at kakayahan sa pagtuturo ng science and mathematics. So, again, maraming salamat po at sana itong programa po ay magtuloy-tuloy at hanggat sa mapakinabangan po ng ating lahat ng mag-aaral dito po sa Bangsamoro. Maraming salamat. Assalamualaikum warahmatullahi wabarakatuh.
Okay, maraming salamat po 𝗕𝗀𝗏 for sa message po on behalf kay Director General Salik. All right. Now it's time for us to open yung floor para sa ating mga questions and answer portion. For the first speaker, may natutunan ba tayo sa ating first speaker? Can you give me a thumbs up there or a heart? Okay. Sige. And I'm sure yung mga audience natin ngayon is excited ngayon, no, na magtanong. And mag-engage para dun sa ating topic natin regarding sa molecular biology. And again, if you have any questions, just type them dyan lang sa chat box po natin, and meron po tayong Q&A portion po dyan for our chat box sa Zoom. And we will be selecting the questions po, no? And so don't miss the chance po, no, na maka-interact natin yung first speaker po natin na si Miss Alicia Aquino. Okay? Sige po. Are there any questions po? And for also, you can also raise your hands po, no, if meron pa kayong questions for the first speaker. Yun. Sige. Siguro ako yung unang magtatanong, no. Ito pala, meron tayo yung first question dito. Galing sa isang anonymous attendee. So hindi siya nagpakilala, no. So ang question dito is, there are possible trainees that didn't make this webinar. Pwede rin po ba sila maka-join doon sa ating upcoming training for our molecular biology training? Ma'am Alicia, pwede ba?
Hello?
Siguro, yes po. Ay, sige po. Pwede po natin sagot po yun, sir? Ay, ma'am?
Yes, they can still join the training. Makipag-connect lang po tayo sa aming project officer and the training coordinator for PGC, Ma'am Eddie.
Okay, sige. I would like also to add that, no? Yes, kahit actually on this upcoming trainings po natin, no, for specifically for BARMM, there are on the first phase, meron po tayong I think 24 participants na selected by our MBHTE. But some of the our educators and also our researchers here is pwede rin sila mag-join sa atin, no, sa ating mga trainings. So we have so many trainings, and we are planning next year to make it on a monthly basis po yung ating introduction to molecular biology, and as well as po yung ating introduction to bioinformatics, no? And for more information, you can contact us po dito sa PGC Mindanao, and later I think we will flash on our screen yung mga contact details po natin, as well as yung mga email addresses natin. Okay? Sige po. So for our second question here is, Ma'am Alicia, we have a question here. Can you simply define what is omics po, no? Okay.
Oh, that's a good question. So omics is many technologies that we can use to study the different levels of genetic information that we have. So kaya siya genomics kasi these are the technologies that we use to study the genome or at the DNA level. Meron din tayong ibang levels such as at the protein level, so that is the proteome. So we have another set of technologies that can be used to further study the proteome. Proteomics po ang tawag doon. And metabolome, that is the environment ng ating perhaps siguro sa intestine. So meron tayong environment doon ng mga metabolites. So that environment is the metabolome, and we can use technologies to study the metabolome, and this is called metabolomics, and so on and so forth.
All right. Okay, sige. Thank you so much, no, for that question. I mean answer, Miss Ali Aquino. No, I think omics is parang ano siya siya yung makabagong teknolohiya ngayon na kung saan yun nga, DNA na tayo, no? So maraming, and ito yung mga makabagong teknolohiya kung saan paano natin i-analyze yung ating mga DNA o iba't ibang mga forms ng DNA natin tulad ng RNA pati yung mga protein natin. And to answer more of that, pwede po natin siguro doon sa ating mga other speakers po natin, no, para mas malaman natin kung ano yung mga natin, yung pwede nating ma-apply dun sa omics natin, no? So may mga iba't ibang field of studies and research here na pwede natin ma-apply po yung omics, no? So okay, sige po. I think we can move on to our next speaker, no? So again, thank you so much po, Ma'am Ali Aquino, for giving us the introduction to ano yung molecular biology. Okay? So next po, for our second speaker who will share to us kung ano yung mga application ng omics, especially yung molecular biology doon sa plant studies. So let me introduce to you our second speaker. So Ma'am 𝗨oc𝗹𝗲𝗰𝗂𝗋 is a Philippine Science High School Southern Mindanao campus alumna with a BS Biology degree from the University of the Philippines, Mindanao. And she has a Master of Science degree in Plant Genetics and Crop Improvement from the University of East Anglia, United Kingdom. At present, she works as an assistant professor in the Department of Biological Science and Environmental Studies here in the University of the Philippines, Mindanao. And she also serves as a technical consultant of the Philippine Genome Center. With the department research strengths on the diversity and as well as on molecular biology, she has some research projects and as well as naging project leader siya, no? Or maybe some project staff in some of the researches that fall under the plant science, especially on the propagation, biodiversity, and the molecular barcoding. So she is also recently interested in bioeconomy using native or endemic plants, and so also some stress physiology and sustainable agriculture. Furthermore, she serves as a journal reviewer on selected local and international journals, and she is also part of the editorial team of the UP Mindanao's Banwa Journal and the Open Access Journal of the Plant Health Research. And her other affiliations include the membership of the Crop Science Society of the Philippines and the National Research Council of the Philippines. So, ladies and gentlemen, let's extend our warm welcome to Ms. 𝗨oc𝗹𝗲𝗰𝘂𝗹𝗿. Take it away, ma'am.
Thank you very much, Don. Please give me a few seconds to share my screen. All right, so subukan po namin i-simplify or at least give you an idea of our studies and ano yung applications niya. I think one of the questions po na lumabas kanina sa question and answer is, ano yung pinakamagandang field to really study to study omics perhaps. And I think marami po kasing applications sa various fields. So for this morning po, I'm going to share with you ano yung applications niya when it comes to plant studies. So if you can hear me well, please give reactions doon sa ating sa inyong link. All right, thank you very much. So I am Assistant Professor 𝗨oc𝗹𝗲𝗰𝘂𝗹𝗿 and my colleagues call me Ma'am Zay or Zay. I am from the Department of Biological Sciences, but I also work under the Science Education and Training Division of PGC Mindanao. So for the outline of my presentation po, we will look at ano yung challenges in plant identification, what is DNA barcoding, general description lang, yung general workflow, what we do in the lab, what is the story of our project, the MIB Davao Bioresource, and ano po yung aming collaborations that arose from this project. So kapag pumupunta po tayo sa gubat or if we are not plant enthusiasts, parang malamang hindi po natin kilala ano yung species ng plants that we have. Generally because hindi naman po lahat ng tanim na nakikita natin sa labas ay meron na siyang bulaklak or meron siyang bunga. Or yung iba naman, they are treated as weeds, mga sagbot as we call it in Bisaya. So if for example, meron tayong mga plants na wala siyang flowers, wala siyang fruits or seeds, minsan or kadalasan, nahihirapan tayo mag-identify at the species level kung saan ba talaga siya nagbe-belong. Also, if we are looking at natural forests and gusto nating tingnan kung ano ba yung identification ng tree na yon, isang problem is if it's a very tall na tree, hindi natin basta-basta hindi tayo nakakakuha ng dahon or ng kanyang mga flowers or fruits. So, minsan or kadalasan, konti lang yung sample na meron tayo for identification. Another one is kapag yung ating plant samples ay native or local at wala pang publications about it. So minsan po, mahirap i-identify or alamin yung species ng ating halaman kung walang guide na available or wala tayong botanist na kasama who will gladly identify. And sometimes, bawal din pong i-collect kasi ng maramihan yung ating plants from the wild, lalo na kapag wala tayong permit. So we cannot collect voucher specimens na usually yun yung ginagamit if mag-identify tayo ng plants gamit yung kanyang dahon, bulaklak, and kanyang mga parts. Especially also if meron tayong plants na they are growing sa protected areas ng ating mga forest. So etong mga challenges na ito, pwede siyang tulungan or pwede siyang isolve gamit po ng DNA barcoding. So kapag sinabi po natin DNA barcoding, tinitingnan po natin yung small segments lang doon sa kanyang DNA. So tayo, marami po tayong DNA. Mahaba po yung ating code. Para po siyang letters, yung A, T, C, G, sabi ni Ma'am Ali kanina. However, pwede naman pong portion lang nito ang titingnan natin at ikukumpara natin sa ibang halaman. So dito sa image po na nakikita ninyo, ito po yung agarose gel electrophoresis na nakikita yung picture. So yung bawat pong line o linya na nakikita natin, pwede po siyang barcode, yung parang nagsha-shopping tayo, di ba meron tayong black and white na barcode. Para po siyang ganun yung kanyang silbi. Meron pong specific na arrangement nitong mga linya na ito, depende sa kung anong species ng halaman meron tayo. So, ang maganda nito, hindi mo kailangang alam talaga yung itsura ng halaman or hindi kailangan botanist ka para malaman mo yung iyong specimen. Also, pwede mo din siyang isabay para makatulong sa pag-identify ng halaman kasabay nung dahon, bulaklak, or prutas na inyong na-observe. So generally, malaking tulong si DNA barcoding kapag may mga limitasyon doon sa morphological data na meron ka. So, ito po yung workflow ng plant barcoding. So, meron kang sample, kunyari dahon, tapos, pwede nyo po siyang dikdikin. Pagkatapos, kukunin natin yung DNA. Tapos, pipiliin natin kung anong genes lang, anong segment ng DNA yung gusto nating tingnan at ikumpara. Tapos we will run it in an agarose gel electrophoresis at titingnan natin using sequencer ano yung pattern ng mga letra na meron doon sa genomic information na nakuha natin. At gamit yung pattern na yon para po siyang barcode na pwede nating masabi, uy, yan siya ay ubas, uy, yan siya ay mangga, and so on. So, para po sa amin sa MIB Davao Bioresource, so ang study po talaga niya ay molecular identification of biological samples na mahahanap sa Davao region. And ang goal po namin is yung aming samples na makukuha dito sa Davao region, pwede namin siyang ma-identify or mapangalanan gamit yung molecular biology na tools. So, also, it will touch yung ating sustainable development goals on life on land, and dahil may samples po kami na meron ding marine, it will also touch yung SDG 14, life below water. So yung flow niya lang po, meron tayong ability or isang lab na tutulong sa atin para makuha yung barcode, i-update natin yung available na inventory online, tapos gamit yung aming faculty who are into communication, tutulungan po kami gumawa ng posters and videos para ma-spread yung awareness of this technology. And at the same time, yung aming results po, pwedeng makatulong if kailangan bang is-conserve, i-propagate, or gamitin yung mga plants na na-identify namin. So yung sa information and education campaign po, we were invited po sa on live talks, and this is with Ma'am 𝗪𝗓𝒿, we talked about yung project po, and through the help of the CHSS team, we were able to create po yung posters. So yung aming team po ay isang napakamalaking team. Meron po kaming from the Biological Sciences and Environmental Studies. Kasama po dito si Chancellor Morao. And then we had the Food Science and Chemistry, si Ma'am Villame, Ma'am Dan. So for the DBSES po, si Ma'am Joan, who is our university researcher, ay kasama din po namin. And for the Department of Humanities, dahil sila po yung magaling sa communication, we have Ma'am Dizon, Ma'am Nidoy, and Sir Villacastin. So after po nun, meron kaming project collaborators, mga students, researchers na hindi pa po kayang i-fund ng kanilang project yung pag-identify ng kanilang samples molecularly. We also had partners with industry tulad ng cacao, banana, and forest resorts. So among our project outputs po, we were able to identify plants found in certain forests na meron po siyang juvenile or bata na plants or mature plants pero wala siyang reproductive structures. So using yung barcode po, we were able to match them kung ano yung species nitong mga halaman na ito. And then another one po is we also tried to see if may relationship ba yung binibenta sa ating nurseries, yung mga cultivated plants na ornamental plants. May relationship ba siya sa mga wild na plants na meron tayo doon sa ating forests. And we were able to match this with the family ng mga monstera. So we also have a lot of other project collaborators, and hindi lang po limited ito sa plants, because aside from banana, cacao, and palm species identification, we also had projects po wherein i-identify namin ano yung disease na meron doon sa crops. So pathogens present in crops. Meron din po kaming partner collaborator na nag-trace po kami kung yung kanilang mga itlog na turtles ay galing ba from same mothers at bumabalik ba sa island yung mothers na yun using the barcodes. And finally po, we also have partners who have fungi and bacterial na samples. So meron po silang in-isolate na fungi or bacteria, pwedeng from soil, pwedeng from plants or other organisms. Tapos using the barcode, in-identify po namin ano yung kanyang species. So medyo napadali po, lalo na kapag hindi ka bihasa when it comes to morphological identification. So I think yan po yung general application ng aming project. And to end this lecture, I just want to say that through this project, natutunan po namin that coming together is a beginning and keeping together is a progress. So working together, tulad ng ginagawa natin ngayong umaga, is truly a success. Yan lang po. Maraming salamat and good morning.
Okay, thank you so much po, no, Ma'am Zay, for your talk. And from that, pwede natin i-open yung ating question and answer portion para doon sa ating mga katanungan para regarding doon sa application po ng omics at pati yung molecular biology sa ating mga plants, no? So, I think we have some questions here, ata. And again, if you have some of the questions, if you want to question it, kung gusto nyo po itanong yon na live, pwede po kayo mag-raise ng hand. Meron po tayong raise hand button po dito sa baba ng ating screen. And doon po pwede po ninyo sabihin po and mag-open po tayo ng mic. At doon po natin itanong yung ating katanungan sa ating speaker. Okay? So, I think, okay. So I think wala namang ata question. Ayun. Meron ditong question ay sabi dito. This is a great field, no, yung plant studies natin. And I think we have so many plants na, if not mistaken, na hindi pa na-identify sa basilan. Yun. Ma'am Zay, if you want to add some of this.
Yes po. Thank you for that input po. Actually po, may partnership din po kami with DOST Grind, yung Grassroots Innovation, and we are coming up with proposals po. So if you are interested to share to us ano yung mga plants na native or endemic sa inyo, and you want us to identify it molecularly, pwede po tayong mag-come up with a partnership on this one, and we can help you identify it po.
Ayun, thank you so much po, no? And I think we have the last question here, no? Second question po dito is what do you do if you find a plant or an organism na wala pang barcode?
Yes
Ma'am?
po. So for now po, ang ginagamit namin is yung universal barcodes lang, yung meron na pong alam natin na code. So meron po kaming instances na wild specimens na wala po siyang kamatch. So dito na po papasok si bioinformatics. So ginagawa po namin, we match it sa saan siya pwedeng mag-belong na family. So we do phylogenetic analysis. So using program po ng computer, ima-match na po yung na-sequence namin na portion ng DNA at ima-match na kung saang grupo siya ng plants most probably mag-belong. Yan po.
Okay, sige. Maraming salamat po, no, Assistant Ma'am Zay, for the second, for giving us a wonderful insights po para sa mga applications po natin ng omics sa plant studies po, no. Sige. Okay po. So may question dito, no. I think may have the last question dito, no. So do you have a training or workshop on how to barcode plants? Yes, we have a training for this and as well as yung workshop, no. Kung saan pwede po tayong mag-contact po sa amin dito sa PGC Mindanao para mas malaman po natin ng mas maayos po kung paano po natin to magawa po yung pagbabarcode ng ating mga tanim. Specifically doon po sa inyong mga lugar. Okay? So again, thank you so much po, Ma'am Zyros, no, for giving us a wonderful message for the applications. No? And now, let's go. The wonders of molecular biology don't stop there, no? So we can explore more of the application ng molecular biology in the realm of the wildlife studies. No? So let me introduce you to the third speaker. So Sir 𝗨𝗪𝗂𝗋 took up a BS Biology at the Leyte Normal University. He also went to the University of the Philippines, Los Baños, to get his master's degree in zoology. And punta din siya sa University Malaysia Sarawak as an attached student. So Assistant Professor Gamalo is currently one of the faculty staff dito po sa amin sa Philippine Genome Center Mindanao. And he is also a staff of the DOST NICER program on applied modeling, data analytics, and bioinformatics dito sa UP Mindanao. He has also authored and also co-authored 10 research studies, tapos nag-co-author din siya ng mga 16 research studies sa kanyang career. He also involved sa different research studies like a humane program funded by the National Research Council of the Philippines, and as well as the biodiversity assessment ng Mount Kampalili project, which is funded by the provincial government ng Davao de Oro. He is also a regular member ng NRCP and member din siya ng IUCN SSC section ng Human Primate Interaction. So ladies and gentlemen, please help me extend our virtual welcome to Sir 𝗨𝗪𝗂𝗋. Sir, your floor is now yours.
Hi, Don. Makita ba yung aking?
Yes po, sir. Makita po. And audible po kayo, sir.
Okay. So thank you very much, no, for that introduction, Don. So if makita na, maybe I can start, no? So again, good morning, everyone. Thank you very much, PGC, for this opportunity. And again, I'm 𝗨𝗪𝗂𝗋. I am a faculty sa UP Mindanao, and I'm going to present this, no, utilizing omics in Philippine wildlife research. So we'll be giving examples from the PGC Mindanao's bat-related studies. Okay, so nasabi na kanina, no, yung maraming applications ng omics, no. So now I will be highlighting yung application niya sa conservation, wildlife conservation, and also the basic research sa wildlife studies. Okay, so let me start by introducing the application of omics to wildlife management and conservation. First, yung ating omics ay pwedeng magamit for surveillance of wildlife and their habitats, no? Particularly yung ating mga wildlife sa Philippines ay paboritong, no, i-trade, no, illegal and legal. So using yung mga barcodes na sinabi ni Ma'am Zay kanina, pwede natin matrace kung saan galing itong mga species na to o yung kanilang mga derivatives. And we can also use eDNA or environmental DNA signature for confirming the presence of cryptic and elusive species. For example, yung ating tarsier, no, na makikita din sa BARMM. I think some parts din ng Leyte, Samar, Biliran, Mindanao, and I think sa Basilan meron ding records. And also, we can confirm, if we can confirm the presence of this cryptic species, pwede din siya magamit for reintroduction purposes ng wildlife, particularly kung siya ay need na i-conserve or i-save. So, second application sa wildlife conservation is detecting hybrids, no? Sa wildlife, isa sa mga naging problema ngayon, particularly yung ating mga species na to na makikita nyo sa screen, itong ating mga crocodiles at itong ating mga warty pigs. These are vulnerable species or threatened species that are threatened because of hybridization. So for example, yung ating mga baboy ramo na mga ecological engineers, nakikipag-breed na rin sila sa mga domesticated animals, and that can actually cause problems. Also, same sa ating mga buwaya, no? Yung ating mga Crocodylus porosus, yung mga saltwater crocodile ay nakikipag-hybridize na rin sa ating freshwater crocodile. And if we have hybrids, ito ay hindi strong, no, sa ating environment at posibleng mag-cause siya ng genetic pollution, no, kung ma-introduce siya sa environment. Okay? And this can also be used to manage habitats of two sympatric species, yung magkalapit. Saan ba i-introduce itong species na to, no? Dito ba sa isang place or pwedeng dito sa isang area? And of course, management of exotic species. So third, conservation application ay ang identification of genetic diversity and adaptation for introduction purposes. So we all know, no, na dapat mataas ang genetic diversity. And if we detect an inbreeding condition, ibig sabihin bumababa ang diversity ng mga wildlife, no, sa ating habitat, ano ang pwedeng mangyari, no? Pwedeng maubos ang wildlife na yon. So if we detect an inbreeding, then pwede tayong magkaroon ng reintroduction purposes. And of course, very important thing, no, to consider if we want to reintroduce is to know if there are adaptive traits na gusto nating introduce sa isang population. No, kung for example, mag-introduce tayo ng something, mga individuals, no? Tapos nag-introduce tayo ng meron silang traits na hindi adaptive sa isang area, pwedeng magkaroon ng problem sa kung saan tayo nag-introduce ng wildlife na yon, no? And for just an information to everybody, if we say reintroduction, the aim of this is to conserve yung wildlife, no? Meron kasi minsan nagagamit siya na, for example, sa mga long-tailed macaques, yung mga unggoy, no? So gusto natin i-move, i-reintroduce sila sa ibang area, kaya nagiging problem na to sa ating area. But that's not the aim of reintroduction. We're just only moving the problem from one place to another. So if we want to answer the problem, dun mismo sa area na lang natin dapat siya gawin. Ang reintroduction ay for conservation. For example, yun nga, yung may mga inbreeding nang nangyayari. Okay, so these are the implications for wildlife conservation. Pero paano tayo ng mga researchers na gusto lang i-answer yung ating curiosity, no? Yung ating mga basic research. Meron bang application ng omics? Yes, meron, no? So for example, sino nakakaalam sa mga hayop na to? These are bats, right? So these bats, particularly itong dalawa, mga harpy bats, harpy fruit bats or Harpionectaris whiteheadi, and this yung sinasabi nating kabog, yung 𝗨𝗏𝗃𝗏. So these are fruit bats, and this one is an insect bat, no? So these bats are flying mammals. Ibig sabihin, they are very important in dispersing our seeds and pollinating our plants, and even controlling our pest insects, no? And it's very important to have a surveillance ng area kung ano ang mga bats, no, na makikita sa isang place. Or di lang bats, or even sabihin natin wildlife. Pero para magkaroon tayo ng surveillance, no, sa ating mga species na to, so ano ang ginagawa kadalasan? Siyempre, hinuhuli yung mga paniki using mist nets. So makita nyo, ito sa ating screen, may paniki na nahuli. And minsan, no, ito ay stressful sa ating mga hayop, no, mga kawawa. So yung PGC Mindanao, nag-ask kami ng question, meron bang way, no, na ma-survey natin yung mga bats na to na hindi sila masasaktan, mga non-invasive way na di na kailangang i-trap? Maybe we can use yung kanilang mga environmental signatures or even their waste, no? So yun, no? Gitry namin i-ask kung pwedeng gamitin yung kanilang mga guano, yung kanilang feces, no? To confirm the presence of this bat species. So ang tanong, gumana ba? Yon, no? So in 2021, nakapublish kami ng paper confirming this, no? Na we can use actually our fecal samples using omics, no, to confirm bat species. So ang ginawa namin, meron nag-study ng bats, no, sa Malagos. And then we collected yung samples na na-collect nila ng bats, and then collect the same from the same individuals. Nag-collect din kami ng mga fecal samples. And then surprisingly but expectedly, no, naka-identify kami using their fecal samples. As you can see here, mga 𝗨𝗏𝗃𝗏𝗃𝗏𝗃𝗏𝗃𝗏𝗃𝗏𝗃, 𝗨𝗏𝗃𝗃𝗏𝗃𝗏𝗃𝗏𝗃𝗏𝗃, these are fruit bats na native sa Philippines, but also present in other countries. However, nung na-confirm namin na itong mga species nga ay present doon using fecal samples, nakita namin na yung ating 𝗨𝗏𝗃𝗏𝗃𝗏𝗃𝗏𝗃, itong blue, ay different sa 𝗨𝗏𝗃𝗏𝗃𝗏𝗃 na makikita sa ibang bansa, itong red. So alam namin na sila ay 𝗨𝗏𝗃𝗏𝗃𝗏𝗃, pero bakit ibang-iba yung kanilang identification? So extend namin yung aming project, and using bioinformatics, ito, no, na-confirm namin na yes, they are 𝗨𝗏𝗃𝗏𝗃𝗏𝗃𝗏𝗃. Ito dito makita nyo, pero our 𝗨𝗏𝗃𝗏𝗃𝗏𝗃𝗏𝗃 in the Philippines is different from the 𝗨𝗏𝗃𝗏𝗃𝗏𝗃 from other countries. Itong violet, no, itong violet, ito yung mga 𝗨𝗏𝗃𝗏𝗃𝗏𝗃 sa ibang bansa. At kung mapansin nyo, ang 𝗨𝗏𝗃𝗏𝗃𝗏𝗃 nila ay mas naglampa sa isang species ng 𝗨𝗏𝗃𝗏𝗃, which is yung 𝗨𝗨𝗏𝗃𝗏𝗃. At sinasabi dito na yung ating species ng mga paniki, nakala natin ay marami lang, no, sa Pilipinas at present din siya sa ibang bansa, they are indeed different na species. Kung makita nyo, naglamp lang ang mga nasa Philippines lang. Same ang nakitang pattern sa isang species na 𝗨𝗏𝗏𝗃𝗏𝗃𝗏𝗃𝗏𝗃𝗏𝗏. Ito yung makikita sa Samal, no, kung nakapunta na kayo sa Samal. At kung makita nyo yung 𝗨𝗏𝗏𝗃𝗏𝗃𝗏𝗃𝗏𝗃 sa ibang bansa, sa Malaysia, Indonesia, ay mas naglampa sa mga rosetos na ibang species kaysa sa ating rosetos, no? So parang sinasabi dito na yung ating bats ay indeed different, no, from the bats na makikita sa ibang parte ng mundo, no? So baka ano kaya ang naging reason dito? Possible ba na talagang different species yung Philippine 𝗨𝗏𝗃𝗏𝗃 at Philippine rosetus? Or sadya lang dahil sa ginamit namin na sample, no? Kasi again, remember, ang ginamit namin na sample ay fecal matter. And ang fecal matter, sila ay yung samples na ginamit ay hindi ganon ka intact, no? So baka mga fragments lang yung mga DNA doon. So ang ginawa ng PGC, no, nagkaroon kami ng meeting at ang possible namin na naiisip, yes, pwede, no, kasi ito yung sinasabi sa ibang bansa na iba ang Philippine population, pero baka dahil din barcodes lang ang ginamit namin, no? Lalong-lalo na ang ginamit pa namin na sample ay feces lang. So maybe maging iba yung kanilang result, magkaiba yung result kung short fragment lang ng DNA ang ginamit, which is yun ang ginamit namin sa project na yon, at iba ang sasabihin kung longer na yung fragments ng DNA. So anong ginawa namin? Naggawa kami ng whole genome sequencing, yung buong genome na, no? Para makita talaga namin kung is it because lang feces lang ginamit or sadyang ganun talaga ang nangyayari na iba yung ating species sa ibang bansa. So ang ginawa namin, nag-whole genome, so nag-extract kami ng mitochondrial DNA sa heart tissue ng same individuals ng mga bats. At then inanalyze namin yung DNA at ito yung naging result. Surprisingly, yes, no? Even yung ating complete na mitochondrial genome na ang ginamit, nagsasabi pa rin na yung ating 𝗨𝗹𝗏𝗃 brachiotis ay different, no? Kung makita nyo, Mindanao, ito yung aming sample. Ibang-iba siya sa 𝗨𝗹𝗏𝗃 brachiotis sa ibang bansa. Same ang sinasabi na yung 𝗨𝗹𝗏𝗃 brachiotis, itong paniki na to, na makikita sa Malaysia, ay mas malapit pa nga sa ibang species ng 𝗨𝗹𝗏𝗃 sa India. At yung ating 𝗨𝗹𝗏𝗃 ay ibang-iba, no, sa mga 𝗨𝗹𝗏𝗃 species, Sphinx at brachiotis na makikita sa labas ng Philippines. And this studies, no, yung fecal matter na ginamit plus itong complete mitochondrial DNA ay sinasabi, no, nagko-confirm sa mga studies na ginagawa din sa UP Diliman at studies na ginagawa din sa ibang bansa. Nagsasabi yon sila na ibang-iba yung ating species. Pero kung i-search nyo ang 𝗨𝗹𝗏𝗃 brachiotis sa Philippines, yung IUCN sinasabi na siya na ay 𝗨𝗹𝗏𝗃 luzoniensis, no? But me personally, hindi ko ginagamit yung 𝗨𝗹𝗃 luzoniensis. Kaya sinasabi din doon na ang luzoniensis ay makikita rin sa Sulawesi. And we don't have any data na mag-support na long luzoniensis ay itong ating species plus yung Sulawesi na makikita sa Indonesia. So again, gusto ko lang sabihin na marami pang questions, no, in terms of the phylogeny, the taxonomy of our bats in the Philippines. So again, no, yun, na-confirm na natin na iba yung ating species genetically and genome-wise. So pero sayang din yung mga samples na na-collect, no? Again, these are precious animals and we want to maximize everything sa na-collect natin na animals. So nung back in 2020, may mga students na nag-research ng same individuals ng bats. Titingnan nila if these bats are infected with blood parasites, no? So na-confirm nila na these blood parasites are Plasmodium. Kung sabihin natin Plasmodium, these are mga malaria parasites na makikita sa mga paniki. Pero hindi nila alam anong klaseng Plasmodium. So ano kayang pwedeng gawin, no, para ma-confirm itong mga parasites ng ating bats? Sige daw, ano kaya? Omics yung answer, no? So ang ginawa namin, no, para ma-confirm, nag-extract kami ng DNA from the liver because part ng cycle ng Plasmodium ay yung liver. And then inanalyze namin din itong mga samples sa ating DNA analyzer. And surprisingly, no, na-confirm namin na yes, yung ating bats na gitry nila using yung blood smears ay merong mga blood parasites. So we use here the primers na ginawa lang para mag-detect ng Plasmodium, ng mga malaria parasites. Primers siya mga DNA bands na ito ang ginagamit para ma-detect ang mga plasmodium na to. Unsurprisingly, hindi plasmodium, hindi malaria parasites yung aming na-detect, which is also a blood parasite but not plasmodium, but is related to plasmodium. We call 𝗵𝗏𝗏. So, yun nga, no, shocking na naman, no, na gumamit kami ng mga primers for looking for plasmodium, pero hindi plasmodium yung aming nakita, kundi 𝗵𝗏𝗏. And this is the first record ng 𝗵𝗏𝗏 sa ating bats na makikita rin sa ating 𝗨𝗨𝗏𝗏 species. And surprisingly, but again, expectedly, no, yung ating 𝗵𝗏𝗏 ay the same 𝗵𝗏𝗏 na makikita sa 𝗨𝗨𝗏𝗏 sa Singapore. Again, yun nga, no, so instead na maraming answer ang na-generate ng research through omics, marami ring problem na na-generate. At ang problem sa research ay hindi talaga siya problema. It's an opportunity to expand our collaborations. And again, sa BARMM is a very important place for wildlife. So these are pictures na nakuha ko, no, kasi kinuha kong consultant sa sa marsh, no, sa Ligawasan Marsh, at marami kayong species na makikita doon, no. So and gusto ko lang sabihin that there are more opportunities to explore. And personally, I study primates, no, and BARMM is a very special place sa akin personally. Ang reason is because nasa BARMM lang na region makikita yung 3 non-human primates sa Philippines. Yung meron kayong unggoy, meron kayong tarsier, at sa inyo lang makikita yung Philippine 𝗨𝗨𝗏𝗏. And again, very important siya, no, kasi may mga studies sa ibang bansa na nagsasabi ng ang inyong Philippine 𝗨𝗨𝗏𝗏 is a different species ng 𝗨𝗨𝗏𝗏. Pero wala pa tayong kalam-alam dyan, no? Pwede natin yan explore through omics. So I think I want to end my presentation doon, no, sa questions and opportunities na pwedeng i-explore ng ating partners sa BARMM. So thank you very much. Yun lang ata doon yung aking presentation.
Okay, sige. Thank you po so much, no, Sir Lee, for that wonderful talk, and as well as yung nalaman po namin yung iba't ibang na-discover po ninyo with the help of omics po, no? And so I think pwede na tayo mag-start sa question and answer natin para matanong po natin yung ano natin gusto natin itanong sa ating speaker ngayon. Sige. So I think meron ditong nag-raise hand, no? So si Sir Peter Jan de Vera, I think pwede po kayong i-type po ninyo yung answer po ninyo, sir. I mean yung questions po ninyo, sir, sa ating question and answer chat box para po para masagot po namin yung inyong mga katanungan. Okay? So yes, meron tayo dito palang comment coming from Omega Joy Shake. So sabi po niya, I am very excited to share all this info and knowledge to my students. But sad to say, due to lack of science, laboratory, and the needed apparatus and equipment, it would be hard for us to teach all of this. And I hope BARMM will consider this situation in the public schools and act on the problem right away. So thank you very much for giving us this opportunity, and we are indeed very grateful, no? So ayun. Sir Leaf, meron kayo may dadagdag doon? So sa mga kakulangan ng ating mga science equipments, gano'n.
Yes, no. I agree, no, and I understand. So yes, no. So talagang isa sa may mga advantage ang omics studies, no. But hindi natin din pwedeng parang itago, no, yung disadvantage niya. At yung disadvantage nito, isa sa disadvantage disadvantages ay ang mahal yung mga gamit sa omics. However, no, I would like to emphasize that parang nakikita na yung omics, no, as a tool, no, to understand yung mga natural phenomena na makikita dito. So ibig ko sabihin, may mga funding na funding agencies na nagbibigay, no, to support this type of studies. So for example, no, sa part ko, just want to share that the UST nagbibigay ng fund to study. For example, yung study ko sa unggoy, no, para lang makita kung ano mga parasites ng unggoy yung nandun. So using omics. So I hope, no, and tama yon, na I hope yung ating government will support us, no, in this itong ating plan, no, to understand yung ating wildlife through omics. Yes, Don.
Okay, sige. Thank you so much, no? So, I think, yeah. So I think, parang nabuhayan tayo, no? Sa nalaman natin ngayon, ng bagong kaalaman kung paano po natin ma-apply yung omics sa ating mga wildlife research, no? And
At
I hope,
Don,
yes po.
gusto ko lang sabihin din, no, na collaboration is also a good thing, no? If, for example, you have the resources, you have the bioresources, and PGC Mindanao, meron silang yung mga resources for omics, maybe we can explore, no, collaboration. But not only, hindi ko lang sinasabi na i-limit lang natin yung ating collaboration dito. Pwede pang iba, no? Pwede tayong mag-extend sa maraming collaboration at maraming opportunities ang ating biodiversity in terms of research.
Okay. Maraming salamat po sa inyong talk po, no, Sir Leif, for giving this wonderful opportunity and talking to us kung ano po yung applications ng omics sa Philippine wildlife research. Okay. Sige. Okay pa ba tayo? Buhayin pa ba tayo? Can you give me a thumbs up there?
Ay, Don, merong nag-question. Can you give us
yes.
tips to get funding related to omics in wildlife?
Yeah, sige po, Sir Leif.
Okay, so I want to answer this na lang. Sorry, Don, for the time, no? But this is very important. So it's also one of my adhikain, no, na maraming mag-study ng wildlife in different ways, no? Pwedeng omics or biodiversity. Yun nga na ginagawa. So yes, a good tip here, no, if we want to study wildlife. Ang wildlife kasi ngayon ay hindi ganun pa ka-sexy, no? Hindi pa siya katulad sa mga health research. But, no, if you can have a collaboration between wildlife tapos health research or even climate change, no? Malaki ito at ito yung gusto ng mga funding agencies, no? For example na lang, no, gusto ko mag-study ng unggoy. Pero sino ba yung funding agency na gustong mag-fund ng unggoy, no? So ang ginawa namin para magkaroon ng funding is gi-relate namin yung aming mga long-tailed macaques, yung mga unggoy, sa mga zoonotic parasites na pwedeng mapasa ng unggoy sa tao or tao sa unggoy. So I think, no, yun ang naging way namin, no, para makakuha ng funding agency. Yun, pwede yung gawin yun, no? Collaboration, mga multidisciplinary collaboration, yun nga, yung pinakamaganda ngayon, no, na explore wildlife and climate change and wildlife and health.
Okay, sige. Maraming salamat po, no, Sir Leif, for that. And yeah, I think we can move on to our fourth speaker, no? From plants and we go to wildlife, then paano naman paano po natin ma-explore pa natin yung yung application ng molecular biology, but this time it is in the realm of the animal health naman po tayo. So let me introduce first yung ating yung speaker po natin ngayon. So Mr. Mijares is a dedicated professional with a rich background in veterinary medicine, no, and as well as academia. So currently, he serves as a faculty member ng University of the Philippines Mindanao in the Department of Biological Sciences and Ecological Studies, or DBSES. And he also brings us a wealth of knowledge and expertise in applying the molecular biology sa animal health. So he graduated with a Doctor of Veterinary Medicine sa University of the Philippines and sa College of Veterinary Medicine sa UP Los Baños. So Mr. Mijares has honored his expertise over the years, and his commitment to animal welfare extends beyond academia. And he also served as a veterinarian sa Animal Solutions Veterinary Hospital dito po sa Davao City. And addition to his contributions in the field, Mr. Mijares has a robust history ng sa corporate sector. He has also worked in operations departments ng KGB Philippines, formerly named as Infon XX, with a focus on directory assistance and services in the US and UK. So apart from his professional achievements, Dr. Mijares is also actively involved in various organizations like the Philippine Veterinary Medical Association, the UPLB Alumni Association, Venerable Knight Veterinarians Fraternity, and the UP Painters Club. So as we delve into today's discussion, we are privileged to have Dr. Mijares, who shares us the application of molecular biology in animal health. Please join me in welcoming him to our webinar. Sir, good morning.
Morning. Narinig niyo ako? Can you hear me?
Yes po, Doc. We
Okay
can hear you po, loud and clear.
So, thank you for the introduction. Ano ko na kung maka-share ako ng screen. So, kita na yung slide.
Yes po, sir. Kita na po yung screen po ninyo, and waiting for a slideshow na lang po.
Slideshow.
Yes po.
Okay. So, ang medyo hindi ganun ka impressive yung aking graphics, no, as opposed to yung previous na presenter natin. Ang major na kasi natin with the omics in applications in animal health is medyo broad siya na topic, no? Malaking kasi on par na rin siya with our sabihin nating on par na rin siya with the medical field, no, in human medicine. Almost the same na rin yung levels niya, although yun nga medyo pagdating kasi in the animal health or the veterinary profession, a lot of times since konti lang din ang veterinarians in the Philippines relatively, no, as opposed to say medical doctors, a lot of times yung focus namin ay medyo nandun sa applications of these parang omics, no, instead of yung pure study itself. Most of the time ang nangyayari, ang mga veterinaryo ay nakatutok sa paano natin gagamitin itong results ng research using omics para matugunan yung mga needs na meron sa ating society, no? So a lot of times hindi masyadong kilala ang omics itself doon sa ginagawa for the industry itself, no? So medyo merong konting disconnect a little bit, although right now talagang nagkakaroon na ng connections within this, and it is a very good development para makita natin kung paano natin magagamit ang omics doon sa pagpapalaganap ng better na animal health, no? Okay, so yun lang yung aking preface muna doon sa ating discussion today. As has been mentioned, ako ay veterinaryo, and although recently lang ako pumasok sa UP, and then naging associated din with the PGC. I think I joined a year ago lang, no? So we haven't had too much of a parang work done, although I know that PGC already has so many projects that will have an impact on, which we'll also understand later, no? So pasensya na kung medyo hindi siya masyado pang in-depth, because we are just starting. I am just starting with PGC. So medyo recent lang din ako. So ine-explore lang din natin muna yung mga possibilities na mayroon sa omics. So ang first natin na parang kailangan ma-establish, ano ba ang ginagawa ng mga veterinaryo? What do we do? And paano namin ito magagamit or itutuloy into the field of omics? So the first thing is, ang pinaka-primary na alam ng lahat is, ang veterinaryo, we prevent diseases and treat sick or injured animals. And a lot of times, ang contact natin with veterinarians as people is usually may alaga tayo na domestic pet, and dinadala natin siya doon sa veterinaryo. So yun yung pinaka parang touch point natin, no? Pagkakakilala natin sa veterinarians. But a lot of times we sometimes forget na ang veterinaryo din is medyo malawak din yung kailangan niyang gawin. Una, kailangan niya rin mag-prevent ng spread of diseases from animals to man, and vice versa. Kaya yung na-mention ni Sir Leaf earlier na merong mga zoonosis, yun yung isa na kailangan din namin ma-address, no, ng aming profesyon. And doon pumapasok ang malaking part ang genomics and the other omics, because meron tayong mga sakit tulad ng rabies, meron tayong sakit tulad ng foot and mouth disease, meron tayong mga like yung avian na mga sakit din, and ASF. So all of these have an impact sa society, and partly tulong ng omics yan is paano natin ma-identify itong mga sakit na to, and maybe with further studies pwede tayo makagawa ng paraan para mapigilan na kumalat itong sakit na to. So isa rin yun na parang ginagawa ng veterinaryo, syempre sa tulong din ng ating medical doctors and other institutions, in order to prevent the spread of these diseases and to understand din paano nag-work itong mga diseases na ito. Another job of a veterinarian would be to improve the lives of animals, keep them healthy. Hindi lang yung ating inalagaan ng mga pets, but also yung mga ginagamit natin for pagkain, like sometimes we have our goats, we have our cattle, we have our chickens, no? So lahat ng ito importante rin doon sa na job ng veterinaryo. Kaya nga medyo konti lang din ang nakikita na veterinaryo doon sa mga clinic, because maraming veterinaryo na pumapasok din dito sa Department of Agriculture and other para matulungan yung mga pag-produce ng ating pagkain, animals for food, kasi importante din ang veterinaryo para magkaroon tayo lahat ng food security, kasi mahirap na magutom talaga ang ating mga kasama sa buhay, no? So those are probably the primary focuses of veterinarians when we do our jobs. So paano natin ito ma-relate ngayon sa omics? So ano ba ang omics in veterinary science? Of course, yung varied and growing na omics fields kasi dumadami ang ang omics fields habang tumatagal kasi nakikita ng mga researcher and mga scientists na parang merong merong category na kailangan natin mapuhunan and cover, and the omics fields actually grows no, dumadami sila. So ang purpose, ang omics sa veterinary science, a lot of times, nagbibigay ito ng tools and yung knowledge para magampanan ng veterinaryo ang trabaho niya. No? So, although, a lot of times yung veterinaryo, medyo, lalo na yung mga matatanda na veterinaryo, sometimes medyo narrow-minded sometimes. But because pumasok na itong omics, natututo na tuloy kami na tumingin, kailangan pala natin malaman bakit nangyayari yung isang bagay at hindi lang para paano to gamutin at paano to ma-stop na mag-spread. Kailangan pala natin maintindihan din kung bakit siya lumalabas at ano ba ang mga factors na nagko-cause nitong paglabas sa itong mga problema na ito. And the way to do that is using omics in order to uncover, kumbaga, yung mga misteryo pa na meron doon sa biological systems na meron tayo. No? So to give a few examples, meron tayong genomics. No? So ito ang isa sa siguro umbrella term no, ng simula ng pag-aaral. So ang genomics, which studies nga yung genes and yung genetic associations na lumalabas yung phenotypes of interest, no, yung mga traits na lumalabas doon sa pag-express nitong mga genes na to, ay importante, of course, para sa animal health and husbandry, meaning paano ba natin mapapabilis ang laki, paano ba natin maaalagaan ng maayos yung ating mga animals, and of course yung sustainable breeding programs. Kunyari, meron tayong goats. Ang gusto natin, yung goats natin ay mag-produce ng mas maraming milk. So, pwede tayong magtingin ng ano ba ang mga breeds na nagpo-produce ng magandang milk. Tingnan natin ano bang mga genes meron dito at baka pwede natin ma-improve yung ating Philippine native goat na ginagamit ng ating mga farmers doon sa ating lugar, sa BARMM and sa other areas. So with this, understanding kung paano nagwo-work itong mga genes para makaproduce nagkakaroon tayo ng sustainable breeding programs, gumaganda yung ating mga inaalagaan na hayop, and natutulungan natin yung mga communities na medyo hindi masyadong maganda yung kanilang stock, pwede natin pagandahin because na-understand natin by the use of genomics kung paano pa itong lumalabas itong mga traits na maganda sa hayop. Next, of course, isa ring important na part is paano natin mas maintindihan yung pag-spread ng mga sakit and paano nagkakaroon ng resistensya itong mga kakaibang mga sakit na to or biological na mga like say bacteria and viruses, paano paano nila na-spread yung kanilang sakit and paano natin mapipigilan yung pag-spread nito. No? So, important ang genomics doon kasi doon natin ma-identify ito ba, kahilera ba to ng a specific type of bacteria or maybe it's a different bacteria and kailangan iba yung therapy na gamitin para mapigilan ito. No? So, isa yon na way para magamit natin ang genomics. Another is of course yung host and pathogen na interaction. Kasi syempre, meron kang say tao, iba naman yung tao sa sabihin mo baka, pero bakit yung sakit ng baka pwedeng matransfer sa tao? So kailangan din natin malaman yung mga yun para magkaroon tayo ng ways na mapigilan yung transmission na yun. Kasi syempre, ayaw natin lahat magkasakit and ayaw natin yung mga kababayan natin magkasakit. So that's one aspect of genomics, how genomics can actually help us to become more aware of how natatransmit itong mga sakit and paano natin mapipigilan talaga. So next, we also have metabolomics, which na-mention na previously with our previous na mga nag-speak. ang mga batikan natin na nagsalita previously. So with metabolomics naman, you are actually going into yung metabolic processes na nangyayari sa healthy and sick animals. And yung relationship ng healthy and sick, paano natin makita and then paano natin mapigilan itong mga problems na to. So with metabolomics, we actually expand yung knowledge natin about general physiology, which impacts everyone kasi lahat ng tao may physiology, lahat ng hayop may physiology, and any little bit of understanding that we get about the physiology will help us to actually develop more programs para ma-keep natin yung healthy na physiology as opposed to may sakit. No? And of course, merong advancements in medical practices. For example, natututunan na natin na yung normal na dextrose na binibigay sa ating mga hayop ay pwede palang iba-ibahin natin yung composition nito para mas maganda yung effect doon sa tao. So, doon pumapasok metabolomics kasi ang tinitingnan niya is yung exactly nangyayari within a certain period of time. Hindi lang as a general whole, but as doon sa nangyayari in real time. So pwede kang maggamit ng tools na meron sa metabolomics para ma-understand itong mga bagay na ito. Another is of course yung production ng new drugs. If we know how the metabolic processes work, kaya tayong gumawa ng bagong drugs. So meron na itong pasok sa industriya ng pharmacy, sa industriya ng chemistry. Lahat ng ito is maintindihan natin or mapagdudugtong-dugtong natin by the use of the metabolomics. Another example would be proteomics, wherein ang tinitingnan naman natin dito is yung protein interactions na ano na nangyayari dito. So malalaman din natin ano ba ang normal biology nitong ating mga animals, bakit sila nagkakaroon ng sakit, and makikita rin natin, ma-identify din natin kung ano-ano yung mga problems na nakikita. For example, dito sa PGC Mindanao, we just became accredited na pwede kaming mag-examine ng samples from animals na merong ASF, and then ma-identify namin sila, and that will help us prevent the spread of ASF, which has been destroying the economy and yung lives ng maraming mga farmers. Yun nga, unfortunately, rabies din, no? Para malaman natin kung may rabies ba yung aso o hindi, and that impacts our animal health and yung health din ng mga tao kasi zoonotic ang rabies. And that can also help us identify kung meron nga bang rabies o hindi itong mga animals na nakasalamuhan natin. So there are a lot of applications, and yun nga lang, no, ang veterinaryo kasi, since sila yung nakakaharap ng may sakit, a lot of times it takes time to trickle down itong mga produkto ng research on omics papunta doon sa ating actual na ginagawa. No? So, yun yung gusto natin ma-bridge rin na gap. Kasi maganda nga, gumagawa tayo ng research and everything, but we also need to understand na paano ba natin ito magagamit para maging maganda yung buhay ng tao and everything. So, it's one thing to consider then na pure research may be a good parang starting point, but we also need to understand na yung research natin dapat may purpose and dapat meron tayong application doon sa research na yon eventually. No, hindi naman kailangan kaagad kasi kailangan muna natin mag-gather ng data, but eventually ang endpoint natin is ma-further yung science and magamit natin para maging mas mabuti ang buhay ng tao at ng hayop. No? So in summary, because the omics disciplines allows us to understand the biological molecules that drive the structure and function of our organism, itong omics talaga ang ginagawa niya is it makes us veterinarians better at the jobs that we are actually supposed to do. So the more na maintindihan natin ng mga to, the more na makakatulong tayo doon sa ating mga animals at yung ating mga tao and society in general. So basically, yun yung aking parang gustong ibahagi, and hopefully in the future we will have more parang projects na pwedeng ma-present sa atin para makita natin kung ano ang gamit nitong omics doon sa animal science and animal health. Okay. Thank you.
Okay, thank you po, no, Dr. Mijares, for that wonderful talk, no. And yeah, marami tayong natutunan on what you have give us on the applications ng animal health, which is number one, pwede po siyang gamitin yung omics para i-prevent yung mga certain diseases na pwedeng makapasa sa atin dito sa as humans, no? Pwede din yung mas malaman pa natin ng maayos kung ano po yung nangyari sa mga certain na mga sakit na nangyari doon sa mga animals, specifically sa mga farm animals natin, mga cattle, mga goats, pati sa mga chickens, no? Ayan. So, I think we can open now for the floor for the question and answer. So, para sa inyong mga katanungan, ulit-ulit po, ilagay po natin sa ating chat box dito po sa Q&A portion para masagot po namin ng maayos po yung mga katanungan. Okay. Okay po. Meron po tayo ditong unang katanungan galing kay 𝗨𝗨𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋𝗋 brucellosis jump from animals to humans? So, and what is the impact of it? Dr. Mijares, pwede po niyong masagot po yung katanungan?
Yes. So although hindi ako masyadong familiar with brucellosis. Ang ano niyan is it's a bacterium, kasi ang brucellosis bacterium yan siya na nakukuha yan from, kunyari nagkaroon ng sakit yung cattle. Natatransmit siya through yung infected na mga blood, yung placenta, yung and nagko-cause yan siya ng abortion sometimes sa cattle, no? So things like that. So ito, ang although ang brucellosis sa humans ay hindi masyadong hindi ako familiar masyado with humans, no, kasi yun nga yung ating jump na sa zoonosis. Most na kaalaman natin is nandoon sa animals mismo, kasi dito siya na-discover sa animals. And like other zoonotic na problems na spread ito because yung contamination noong fluids or noong organs na meron dito sa animal na to ay naililipat doon sa tao, or nahahawakan or nakakain or things like that. And a lot of times this is why yung zoonosis is such a problem, because a lot of times meron tayong hindi natin nalalaman na pwede palang magkaroon ng cross transmission itong mga animals na to tsaka yung tao. So a lot of times hindi yung emerging diseases natin, yung mga problema na nakikita, for example yung COVID, ang nangyari daw kung bakit nagkaroon ng COVID is because merong animals na kinain. And then yun yung nag-source ng virus mismo, and then nag-spread into the human population. So ito rin yung pwede mangyari doon sa ating animals na meron na mismo, no. So yes, contaminated na mga parts ng ating animals, blood, fluid, sometimes yung mga organs na sometimes improperly prepared, or pag namatay yung hayop, kinakatay ka agad, no, nang hindi pa nalalaman kung bakit namatay, and then kinakain, no, worst case scenario, kinain ng hindi linuto ng maayos, ginawang kilawin or whatever na ginawa. And this can cause problems, which is why we need to understand yung mechanisms kung paano nasi-spread ito, and that's where omics comes in. Kasi doon na natin nalalaman ano ba ang meron dito sa mga bacterium na to, or bacteria na to, or virus na to, na nakaka-cause ng cross-species na transmission. So, yun yung kailangan natin na kumbaga maintindihan. And yun yung trabaho ng omics and yung researchers na nasa omics na fields para maintindihan natin itong mga bagay na.
Okay, so maraming salamat po sa inyong kasagutan po, no, Dr. Mijares. And I think isa rin sa would like to also to emphasize din yung isa ding application ng omics sa animal health kung saan let's say meron tayong mga farm animals na pwede natin gamitin yung omics approaches para ma-improve yung mga nutrition ng ating mga animals, mga farm animals. Yun. So, yeah. Sir, pwede po kayo makadagdag po for that? For that po, sir? Yung
Well,
application
actually,
po
ang
natin sa
omics is ang primary, no, nung pinakaunang pasok nitong omics talaga doon sa industriya ng veterinaryo. Kasi una nga hindi pinapansin masyado ng veterinaryo kasi tao yung unang focus ng mga studies. So sa kami sa veterinarians, trickle down effect ito. Yung mga studies na nangyari sa humans, naisip ng veterinarian, uy, gamitin natin na. And yung pinakauna na application na nakita is doon sa nutrition at paano padami at magpalaki ng ating mga inaalagaan na hayop, no? So yung actual industry na gumagamit ng omics a lot of times is ang feed industry. Kasi napakalaki ng industriyang ito, napakalaking pera na meron, and meron talaga silang mga research center para maggawa ng feed formulation, malaman kung paano ang epekto nitong feed doon sa ano. And a lot of research has been actually directed towards animal nutrition and animal feeds. Kasi nandun yung pera, nandun yung malaking bahagi ng pera talaga ng
Yes.
research, no? So yun, isa talaga siya entry point ng omics para sa animal health.
Sige, okay. So, yeah, ako as yung actually previous worker is isa akong taga-check ng quality ng mga animal feeds, no? And one of the important dyan yung feed formulation, kung saan nakafocus sila sa nutrition mismo ng animals, kung paano nila mapalaki yung, let's say, yung sa manok, kung paano mapatigas po yung shell ng itlog ng manok, something like that. So, napakahalaga po ng ating omics kung ma-apply po natin yon at mas ma-improve pa lalo yung nutrition ng ating mga hayop na ating mga inaalagaan. Yeah. So, yeah, so with that po, also maraming salamat po ulit Dr. Mijares for giving your inspirational your experience po, and as well as yung pagbibigay po sa amin ng malawak na kaalaman regarding po sa kung paano po namin ma-apply yung omics sa animal health. Okay? So, yeah. So, thank you po so much, no, sir? So, yeah, I think meron na tayong plants, meron na rin tayong animals. Hindi magpapahuli si food. So, for our I think this is our fourth speaker, no? So for our fourth speaker, our fifth and the last speaker, no, is let me introduce you for that, for giving us the application ng omics naman sa food sciences natin. Okay? So for that is Ma'am 𝗨𝗪𝗂𝗋𝗶𝗆 is an assistant professor sa Department of Food Sciences and Chemistry sa College of Science and Mathematics dito po sa University of the Philippines, Mindanao. She is currently teaching sa different undergraduate courses sa Bachelor of Science in Food Technology program, which also includes food microbiology, food chemistry, and food processing. And she is also handling the advanced food microbiology course sa MS Food Science program. And Assistant Professor 𝗨𝗪𝗂𝗋𝗶𝗆 is also obtained her Bachelor of Science in Food Technology degree sa UP Mindanao. And she also gained her industry experience from working as a quality assurance specialist or analyst sa research and department associate, and as well as technical consultant in some various SMEs sa Davao and Butuan City. And noong 2017, she obtained her master's degree sa food sciences with the minor ng microbiology. And her research is focusing on the centrifuge-free DNA extraction method sa bacterial detection using a DNA-based nanobiosensor. Currently, she is involved as a staff or a project staff in a collaborative project sa MSU Gensan, so which is the project entitled Multidimensional Landscape Analysis of Food-Associated Antimicrobial Resistance. So yung mga antimicrobial resistant na bacteria. So among her specific research interests include probiotic properties ng lactic acid bacteria, some ethnic fermented foods, and as well as yung food product development. She also serves as one of the faculty project staff na under sa Philippine Genome Center Mindanao under the Omics Laboratory Services Division. And without further ado, ladies and gentlemen, please welcome Ma'am 𝗨হি𝗹𝗶𝗰𝗂𝗋𝗆. And I think naka-recorded yung kanyang talk for now, and I think I may share their screen. Sige po, wait lang po. Sharing. Okay, so nakita na po ba yung screen? Can you give me a thumbs up there? Sige.
Hello, good morning, everyone. My name is 𝗨হি𝗹𝗶𝗰𝗂𝗋, and for this morning, I'll try to briefly discuss some of the molecular techniques used in food applications. First, let me define what is food science. Hello, good morning, everyone. My name is 𝗨হি𝗹𝗶𝗰𝗂𝗋, and for this morning, I'll try to briefly discuss some of the molecular techniques used in food applications. First, let me define what is food science. It is a branch of science which aims to understand the physical, chemical, and microbial makeup of food. It is actually a collaboration of many scientific disciplines. We have chemistry, biology, biochemistry, even engineering and mathematics. Meanwhile, food technology is the application of food science to improve the quality and sustainability of food products. So this includes improvement of preservation methods and processing techniques in order to extend the shelf life of food, even the packaging, distribution and logistics, food safety, and assured supply of food. For years, food is analyzed based on the following characteristics: a biochemical, physical-chemical, either physical or chemical. So these are gold standard methods. There are gold standard methods in order to determine or analyze these characteristics. However, these may give us very limited results. For instance, yung physical-chemical properties, maraming parameters under physical chemical. An example is for yung acidity na isang food product. So pH is very crucial in a food product, di ba? Isa yun siya sa mga distinct attributes niya sa food product. Yung titratable acidity or yung total soluble solids. Physical characteristics can pertain to some textural characteristics like hardness, crispiness, or elasticity ng isang food product, or yung viscosity ng isang food product. Pwede rin yung color. Pag chemical characteristic naman, it can also pertain to nutritional composition. Pwedeng vitamins and minerals, pwedeng antioxidant content, protein content, moisture content, something like that. So as I mentioned earlier, limited lang yung results nila kasi yung food industry ngayon, yung global trend, yung global demand sa food industry is not only limited sa ganitong characteristics. Sometimes, they require further tests. Like, example lang, no, yung food authenticity or species identification na isang particular raw material. You have to prove na 𝗨𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏 tuna sample mo or yung tuna na in-export mo sa Japan, for example lang naman. So, these characteristics may not give us the data that we need para ma-certify yung ating food product. In order to augment or supplement our data from those biochemical, physical, chemical characteristics that were mentioned earlier on, we can also apply some molecular techniques in order to understand further the identity and characteristics of our food products. Okay? So when we say molecular techniques or molecular methods, so we analyze nucleic acids of interest, pwedeng DNA or RNA. The RNA is actually a single-stranded DNA, and it's really common on viral samples. Okay, so DNA naman, you can from the food sample mismo, pwedeng yung bacteria present or microorganisms present. Okay, so maraming possible applications ang molecular techniques in food. Okay, so ito yung mga i-enumerate or i-discuss ko briefly in the following. In the slide, will be some of the many applications in food. Okay. First, we have the detection of specific genes, especially for genetically modified organisms. Then we have detection of antibiotics and pesticide residues, kasi these are considered as chemical hazards. Pathogen detection. Okay. It's really important for food safety, kasi we don't want na merong contaminations food product natin that could eventually lead to foodborne illness. Ayaw natin ng mga outbreak na yan kasi it will lead to economic losses and of course lives. So as early as raw materials pa lang sa processing hanggang distribution, the food industry really invests in detection of foodborne pathogens. Molecular techniques can also be used to detect contaminants or adulterations or adulterants in your food product. This is quite related to the next item. We have halal and kosher certification. So dito mas makarelate tayo sa halal certification. When we say halal, so dapat wala haram derivatives, no? Hindi tayo makadetect ng mga metabolites from haram. So some method, yung current method ngayon ng halal is gumagamit sila ng mass spectrometry. So, detect na niya yung mga metabolites, yung mga derivatives from porcine, for example. Yun yung mga chemicals or volatile chemicals, yun i-detect niya. But for molecular techniques, pwedeng directly doon sa DNA mismo. I-detect niya if there is a pork DNA, for example, in your supposed to be halal compliant tuna sausage. Ganyan. Okay? So direkta siya sa DNA. So since we are dealing with nucleic acids, mas accurate siya. Okay? And yes, of course, we can use molecular techniques for species identification, especially if you want to prove na, for example, yung product mo si tuna, for example. If you are selling tuna and ito yung kanya species, no? 𝗨𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏 and you want to prove na authentic yung product mo, then you can subject your sample for molecular identification until the species level para ma-prove na yun talaga ang kanyang species. Something like that. Okay? And of course, this is related also to food authenticity and legislation. So these are just some of the many applications of molecular methods and techniques in food. Okay, generally, when we use molecular techniques in analyzing our food products, depende kung ano yung application natin, as mentioned earlier. So for example, we have a fish samples, fish na lang. Actually, yung sample nyo pwede namang hindi fish, pwedeng yung kahit anong sample, pwedeng plant-based, animal-based, pwede yung mikrobyo na galing sa food product ninyo na na-isolate. Pwede yun. We extract the DNA first. Of course, we have to break open the tissue or the cells so that we will be able to get the DNA. Next, may tinatawag na gel electrophoresis. So this is one of the basic steps sa workflow na ito kasi we would like to know if the DNA na in-extract natin is present and intact. Okay? Present and intact. If it is present, then we can proceed to amplification. Ito na yung familiar tayo, PCR, or polymerase chain reaction. So polymerase chain reaction will amplify your target gene or yung gene of interest ninyo. And then once it is present, then syempre mag-amplify siya. Kung hindi siya present, it will not be amplified. Hindi siya ma-detect ng machine. And yung tawag natin sa PCR product are amplicons. Okay? Yung amplicons na yan can be purified and then sequence siya for identification. Now, yung identification is pwede siyang combination ng yung identification is carried out through bioinformatics. So with the help of computers para ma-deduce natin yung identity ng microorganism. Kasi yung gene sequence ng isang microorganism is medyo mahaba, depende doon sa gene sequence or depende doon sa organism mo. So, hindi natin yun siya kayang imano-mano, but using a computer, okay, kaya siya bioinformatics kasi nag-marry yung biology and computer science para tulungan tayo to process that big data. In pathogen detection in food, there are two major detection methods. We have the culture-dependent and the culture-independent. Sa culture-dependent, sa name pa lang niya, we try to grow the target microorganism. For example, ito yung fish product mo, and yung gusto natin na i-detect na pathogen ay Vibrio parahimolyticus or Vibrio vulnificus, for instance. So, i-grow natin siya sa laboratory in a plate, in a sterile environment. Then we try to detect it using differential or selective culture media. However, there are certain microorganisms na hindi siya tumutubok or hindi natin ma-simulate yung nutrients niya from its natural environment sa laboratory. So we do culture-independent methods. Sa culture-independent method, obviously, we use molecular techniques. Pwedeng directly from the sample, hindi na tayo mag-isolate ng microorganism and then i-grow pa siya sa plate. So we skip the culturing step. Kaya siya culture-independent. So directly from the sample, we extract the DNA and then subject it to PCR, for example. And then we detect the marker genes, yung target gene of interest mo. I mentioned it earlier about polymerase chain reaction or PCR. So I think we are all familiar with this because of COVID-19 pandemic, Kasi isang variation ng PCR ang ginagamit to detect SARS-CoV-2 in saliva or saliva samples or yung sputum samples. So marami siyang variations. Merong endpoint, RT, qPCR, multiplex, and lab. But the most common method is the endpoint detection. Lahat yan sila ay variation ng PCR. They share the same principle. In simplest terms, PCR actually makes multiple copies of your target gene. For example, meron kang fish sample dito, we extract the genomic DNA, the genomic DNA is very large. So we only target a segment of that. So meron kang primers that will only target a specific region of that DNA. For example, yung segment na yon corresponds to the gene sequence that pertains to its identity. For example lang. So pag na-amplify mo yung gene na yon, yung gene sequence na yon or region na yon, you will be able to identify that fish species. Okay? For example lang. Okay? So that gene of interest, ilalagay natin siya sa PCR machine, and then the PCR machine, through varying temperatures, cycles, gagawa siya ng multiple copies of your target gene. So basically, if present yung target gene and optimized yung conditions ng PCR, present lahat ng reagents mo, tama yung procedure mo, and hindi malfunctioning yung equipment mo, then you will be able to amplify or detect your target gene. Pero kung hindi siya present, most likely, hindi siya ma-amplify. So PCR can be used for detection, presence, or absence of your target gene. Endpoint PCR or conventional PCR, ito yung pinaka-common method ng PCR, excuse me. It amplifies a specific DNA sequence using a set of primers that binds to the ends of your target DNA sequence. So it uses varying temperatures. So for example, di ba yung DNA mo, meron siyang complementary. A binds with thymine, adenine binds with thymine, cytosine binds with guanine, so on, and vice versa. So for example, you have a gene sequence that encodes for the Shiga toxin in 𝗶𝗨0157. So 𝗶𝗨0157, makakause yun siya ng hemorrhagic diarrhea. Even 10 cells lang ang nakain mo. So for example, ito yung gene sequence niya. Yung complementary gene sequence niya is ito. So AP, 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 so, and vice versa, so on and so forth. So, kumbaga, kokopyahin natin yung gene sequence na yon and we produce multiple copies para ma-detect siya. This is just an overview of PCRs. You have your PCR components, including your DNA sample or template. Then, i-mix yan siya, i-optimize, ilalagay sa thermal cycler. The thermal cycler will, siya na yung bahala doon sa changes in temperature from as high as 95 down to 55 and 72, which corresponds to the three important steps: denaturation or separation ng DNA, then annealing, o yung pag-bind nila balik, and then extension. And then, ito lang one cycle. So in one cycle, we produce 2 DNA. So from one DNA template, magka-produce siya ng 2 copies. So exponential. So yung pag-increase ng amplicons. So you produce several copies after several cycles of PCR. How do you know what type of PCR method is best? Diba maraming na-mention doon kanina. Yung RT-PCR, qPCR, multiplex PCR. It really depends on the type of microorganism or your target microorganism or your target gene of interest. Also, yung sample composition or matrix. So magdedepende yan siya sa type of sample or yung food product mo. Okay? And then, of course, yung available equipment, available reagents, and the skills or expertise ng analyst. And of course, kailangan din siya i-optimize yung method na napili ninyo doon sa, i-optimize yung method based doon sa pangangailangan.
All right. So, yeah, thank you so much for Ma'am Gigem for that wonderful message sa ating application ng omics in terms of sa food sciences po natin. Okay. So, yeah, well, we have gained some valuable insights into this role ng mga different applications natin like plant studies, the wildlife studies, the animal health, and the food science. So as we conclude yung ating webinar for today, so let us now hear for some of the closing remarks po natin from the OIC of the PGC Mindanao, Sir Dr. Joel Hassan Tolentino. Sir Joel, thank you so much.
Hello, Don. Can you hear me?
Yes po, sir. Loud and clear po.
Madayaw kaninyong tanan. Today is just the beginning of our journey on bringing molecular biology and bioinformatics to the Bangsamoro Autonomous Region in Muslim Mindanao, or BARMM. PGC Mindanao's mission to create awareness and enhance appreciation for scientific tools in molecular biology among the bright minds of BARMM is not just a project. It is a commitment to empowering the next generation of researchers, teachers, and faculty in the BARMM region. This also goes to our partner from the Mintal Comprehensive High School. Through the series of capacity building and awareness activities, we are committed to sowing the seeds of knowledge that will flourish into scientific innovation. I extend my heartfelt gratitude to all the participants, both from BARMM and MCHS. I truly hope you learned something from this webinar. The speakers, who are PGC Mindanao technical experts and staff, and partners who have contributed to the success of this webinar. Your enthusiasm and commitment to advancing molecular biology in BARMM and MCHS are the driving forces that will make this project move forward and achieve its goals. I also thank the Ministry of Basic, Higher and Technical Education of BARMM, which is led by the Honorable Minister Mahagar Iqbal, with his Director Generals Abdullah Salik for the Basic Education, who is represented today by Chief Yul Olaya. Sir, thank you, sir. And DG Marjuni Madi for the Higher Education, sir. Director Michael Gatela of UP Mindanao Pahinungod, and PGC Mindanao, PGC Executive Director Dr. Felicitas Lacbawan for the invaluable support. As we conclude this webinar, I hope that participants have not only gained substantial knowledge in molecular biology and omics, but have become torchbearers, igniting interest and collaboration in omics through research and training extensions. Thank you for being part of this significant moment. Shukran, daghang salamat. Here's to knowledge, collaboration, and a future shaped by the transformative power of molecular biology, omics, and bioinformatics. Maayong buntag or maayong udto sa tanan.
Okay, so thank you very much, Dr. Joel Hassan Tolentino, for that wonderful closing remarks for our webinar. So thank you very much for everyone who joined us today in our webinar, and we hope to see you here in UP Mindanao for future trainings and collaborations. And to get your attendance or the certificate of your attendance here in this webinar, you can click on the evaluation link as shown in the chat box below po, no? We have our staffs here who are sending the evaluation links, and you can kindly click those links for you to have fill up your evaluation form to get your certificate of attendance. Okay, so I hope marami po tayo natutunan, no, in this webinar. And take note, this is only the start of our exploration in our journey in molecular biology. And I think this marks the end of the introduction of the molecular biology and its applications. And thank you everyone who participate and who supported us here in this webinar. And we hope that you are excited as much as we explore the intricacies of this field throughout this day. Yeah, so Again, thank you for joining us and let the exploration begin. So, maraming salamat po everyone.